Have you ever wondered what happens after we eat food that contains fat, protein, and carbohydrates? During digestion and absorption process, fats are broken down into its monomers, amino acid, protein into fatty acid and glycerol, carbs into simple sugars. Amino acid undergoes beta oxidation to generate acetyl-CoA. Fatty acid undergoes oxidative degradation to generate acetyl-CoA. Acetyl-CoA then enters citric acid cycle. Glucose is mainly produced from the breakdown of carbohydrates.
When glucose enters the cell, enzyme that can phosphorylate the glucose, which is hexokinase, and convert into glucose 6-phosphate. Two kinds of metabolic pathway that glucose 6-phosphate can enter, which is glycolysis and pentose phosphate pathway. There are two phases in pentose phosphate pathway, but we look irreversible oxidative phase first. Irreversible means enzymatic is not reversible, and oxidative means involve redox reaction. Redox reaction means substrate got oxidized and coenzyme got reduced. And the second one is reversible non-oxidative phase. Reversible means, for example, ribulose 5-phosphate can converted to cellulose 5-phosphate and vice versa. And non-oxidative means the reaction does not involve redox reaction. Substrate does not got oxidized and non-involvement coenzyme. Glycolysis. Glycolysis has 10 steps. The first 5 steps are the investment phase. Investment phase, total ATP produced is 4. And the last 5 steps are called the payoff phase. Payoff phase, 2 ATP will be produced. Substrate will converted into pyruvate. Net ATP production is 2. Means total ATP invest is 4, so already payoff is 2, means net ATP is 2 because 4 minus 2 is 2.
After the production of pyruvate and there is no oxygen, anaerobic glycolysis will occur, and this will produce lactic acid. This causes muscle cramps that happens after and during exercising. Meanwhile, when there is sufficient oxygen, aerobic glycolysis will occur. The pyruvate will go through oxidation to form carbon dioxide and acetyl-CoA.
Krebs cycles take place in mitochondria of the eukaryotes and the cytosol of the prokaryotes. The acetyl-CoA combines with oxaloacetate to form citrate. The next step, the citrate is isomerized into isocitrate. Next, the isocitrate is oxidized into alpha-ketoglutarate, and in this reaction, a molecule of NAD is reduced to NADH, and a molecule of carbon dioxide is generated. Next, the alpha-ketoglutarate is converted into succinyl-CoA, and in this reaction also, a molecule of NAD is reduced to NADH, and a molecule of carbon dioxide is released. The next step, this succinyl-CoA is converted into succinate. In this reaction, a molecule of GTP is generated. Next, succinate is converted into fumarate. In this reaction, a molecule of QH2 is generated, which is used for production of FADH2. Fumarate is then converted into malate, and in the last step of the malate is converted into oxaloacetate, and this reaction also NAD is reduced to NADH.
NADH and FADH2 from the TCA cycle will go on to be used in a set of redox reactions that transfer electrons in oxidative phosphorylation to generate massive amounts of ATP. Oxidative phosphorylation is also known as electron transport chain (ETC). The goal of ETC is couple energy stored in electron acceptors to a proton gradient that drives ATP synthesis. There are 5 complexes in the inner mitochondrial membrane. From the left is complex 1 to the right till the complex 5. Complex 5 is better known as ATP synthase. There is also coenzyme embedded in the area of ETC, CoQ and cytochrome C. The oxidative phosphorylation is broken into 2 distinct parts. The complex portion from 1 to 4, including CoQ and site C, is known as ETC. This is where electrons get shuffled from one complex to another. The ATP synthase, which pumps the proton gradient to generate ATP, is technically known as chemiosmosis. First, an ADH approaches complex 1. An ADH gives up its protons and electrons and becomes an AD+. In the process, it donates its electrons to complex 1. When the electrons enter complex 1 gets supercharged. When this complex gets supercharged, it has the energy to pump the proton from the mitochondrial matrix into the intermembrane space. After a while, electron will sit in complex 1, and the proton gradient is beginning to form on the top of the intermembrane space. It has much more protons than exist on the bottom of the mitochondrial matrix. Complex 1 will pass its electron to CoQ. The electrons will go to CoQ and sit there awaiting further instruction. Then, FADH2 comes along and approaches complex 2. FADH2 gives up its electrons and turns into FAD. It donates its electrons to complex 2. Complex 2, however, cannot become supercharged and cannot pump protons from the mitochondrial matrix into the intermembrane space. The electron sits in complex 2 and gets passed to CoQ. The electrons are sitting in CoQ before passing to complex 3. When electrons go from CoQ to complex 3, it supercharges complex 3, which creates enough energy potential to pump the proton from the mitochondrial matrix through complex 3 into the intermembrane space. Complex 3 will pass its electrons on the cytochrome C. At cytochrome C, the electrons arrive and then get passed to complex 4. At complex 4, electrons enter it and supercharge. Once supercharged, complex 4 has enough energy to pump protons from the mitochondrial matrix into the intermembrane space. The proton gradient forms. The electrons sitting inside of complex 4 and need to pass to the final electron acceptor, oxygen. The electrons pass to oxygen, which splits into 2 oxygen ions, and protons are added, creating 2 water molecules, H2O. In the complex 5 or ATP synthase, the ATP synthase is going to use this proton gradient to generate massive amounts of ATP. ADP wants to turn into ATP, which is a higher energy molecule that can be used to give energy throughout the body. Proton flow from the intermembrane space down through ATP synthase back to the mitochondrial matrix. There is an energy input that catalyzes the conversion of ADP to ATP.